Welcome to Screens of the Stone Age, the podcast where scientists review movies about prehistoric people. My name is Josh Lindell. I'm a grad student studying Neanderthal teeth, and I'm here with...
I'm Dr. Kimberly Pomp. I am a bioarchaeologist. I study the human skeleton, health and disease, and evolution.
And I'm Dr. Ross Barnett, paleogeneticist interested in extinct Ice Age megafauna.
And today, in honor of the recent de-extinction of the dire wolf, we are reviewing a movie called Dire Wolf from 2009, although this movie also aired, I think, on the Sci-Fi Channel under the name Dino Wolf.
Oh, that was the same one? I was going to put that on. I was like, I kind of want to watch Dino Wolf instead.
It's the same movie, just a different name. Who wants to summarize Dire Wolf for us?
I don't, but I
Ross
can.
has to.
Yeah. Okay, so strap in, everyone. I mean, this is another one of Josh's choices, which, yeah, I mean, there's the bottom of the barrel, and then there's like, you're actually into the wood that the barrel's made from. That's where you're scraping. It's so Dire Wolf. Okay, so it opens on Clark Biogen, which looks like it's been made from CG on a N64. I mean, the CG in this film is terrible. But Clark Biogen, and we see, if you're in the know, as I am, a replica of a dire wolf skull, which you can get from the company Bone Clones for, I think they sell for about $300. And we meet a blonde scientist whose name, I don't know, it's not important, but I think she's called Jamie, and she says that there's a door sensor has gone nuts, and she starts flirting with the really quite not attractive security guard. He's like a schlubby, 50-plus security guard. But she has this weirdly flirting kind of conversation with him while she's reading a folder from her filing cabinet about bone density, which is not important to the plot in any way. But she sends this security guard that she's been flirting with, who's called Ed, to search for what's going on with the door sensor. And because it's a state-of-the-art biogen facility, everything is totally dark and industrial. It looks like the inside of the Nostromo from Alien. And Ed goes and he sees that there's a pod door open, which is covered in sticky goo. I mean, it looks like the transporter from Cronenberg's The Fly. That's the kind of aesthetic they're looking for. And it's got goo all over it. And just as he sees that, a creature appears behind him and rips him to shreds. So that's not great. And the blonde scientist, Jamie, she hears Ed screams and gets scared. And so she actually makes the logical and sensible choice of locking what looks like, what would you call them? Like Citadel bank vault doors that are all over this place to kind of protect her from the thing that has escaped. But, oh no, look, it's behind her. And so she absolutely gets destroyed by this creature as well. And then we cut to an all-American diner, and we meet Sheriff Parker, who is the local sheriff, and Jenny, who is a central casting ginger waitress, who is there to kind of introduce the sheriff, who is signposted as being very much on the spectrum. He likes a round-the-clock breakfast, where all of the breakfast items have to be at particular hours of the clock. So that's great. He's a bit of an asshole. That's the impression we get right from the start.
Mm-hmm.
The game warden comes in, who's a young, chiseled, good looks, young blonde guy who's called Jim, I think. Correct me if I'm wrong. He's the game warden. But it also turns out when he's talking to the sheriff that the sheriff is his foster parent. He was fostered by this weird, reclusive OCD individual who is the sheriff. But he's turned out okay. Jenny, the waitress, likes him. We also see at the bar, at the diner, Amber, who is Jim's ex, and her boyfriend, Billy Ray, or Billy Roy, I'm not sure. Billy Ray Joe Wayne. I mean, just a total hick douchebag.
All of whom are blonde. They
Yeah.
keep introducing characters for the first half hour. There's like 24 characters, and about 3/4 of them are blonde for some reason.
Yeah. Jenny's a redhead. Ed, I mean, it's hard to tell what color his hair is because he's in the biogen. I think he's balding and dark-haired. Yeah, and we find out some kind of facts that don't really matter, but Jim, who's now the game warden, his office has been vandalized. They don't know who it is. There's some assholes at the diner, who give him a hard time and say that Jim's dad shot his mum dead. That's why he was an orphan. That's why he's been fostered by the sheriff. That's why he doesn't like guns. So he's a game warden. He doesn't like guns. He prefers knives. In the words of Lockstock, guns for show, knives for a pro. So that's about everyone introduced pretty much. Apart from there's an army general who is being informed that there's been an accident at the lab at Clark Biogen. So somehow the army are involved with this fuck up that's happened. Okay, so then we cut to Devil's Canyon, which is where Jim works. We see a hunter who seems fairly inept. He's like stalking a young deer, and he says very loudly, "Hello, Bambi," just as he's about to shoot it. Now, I don't know much about hunting, but talking out loud when you're close to your prey seems like a bad idea. Anyway, he gets dire wolfed straight away. He gets like mauled, so that's not good. But he manages to sort of let off a shot as he's being killed, and the sheriff hears the gunshot. So that sort of starts things off. Somehow the monster has escaped. And it's really much, it's a game of kind of Monster of the Week bingo from this point onwards. It has all the cliches all the time. Yeah. Amber and Billy Ray are out camping. Jim is who also heard the gunshot. He's looking for what caused it. He finds the dead hunter. That's not great. So now we've got set up all the conflict. We've got the monsters on the loose. Amber and Billy Ray are out camping close to where the monster is. We've got Jim and the sheriff out searching for the monster. And we've also got some agents, FBI, I think, who've been sent by the army to find out what's going on at Biogen. First, they find bloody handprints all over the door. That's not a good sign. There's a dead security guard just outside the door, so this explains how the animal escaped. The security guard came to kind of take over his shift and escaped straight away. The FBI agents go in, they find some survivors. So there's Dr. Renault and Tina. So Tina is a scientist at Biogen. Dr. Renault is the kind of leader. Tina shows the agents the experiment. But, I mean, they're skirting around what it is. But clearly, given that we know the name of the film is Direwolf, it's a direwolf that they've somehow bred for reasons. We see a mom and a kid camping. The kid spots the direwolf in the bushes, makes friends with them, tries to offer him a hot dog bun. We kind of expect that they're going to get horribly mauled, as everyone else has, but somehow they don't. We get a lot of exposition back at Biogen from Tina. She says that they have Canis dirus, the largest canid ever, the most common fossil in Rancho La Brea. 3,000 individuals have been found. We have that it coexisted with man. There's lots of discussion about whether it's a predator or a scavenger. That it went extinct so recently that it's hardly a fossil at all. And they've created a direwolf hybrid in Clark Biogen. And then one of the agents says, "So you've made the hybrid with what? With a grey wolf?" And Tina says, "No, with human stem cells and bone marrow from the direwolf." It's like, oh, okay, great. So you've essentially created a werewolf. So that's cool. Cut back to Amber and Billy Ray. They're having relationship problems based on the fact that Amber has no personality and Billy Ray is a total douche. They're kind of, I don't know, they're just not compatible in any way. She's on the rebound from Jim, I think, and Billy Ray was the closest available male in their tiny one-horse town. The sheriff turns up at the Biogen company. There's a standoff with the FBI. they talk about how they've got an escaped lab animal, and the sheriff says, that didn't look like the work of a guinea pig. Billy Ray gets eaten at the campsite. Amber runs away. She runs for, it feels like, forever. She meets a fisherman. Obviously, the fisherman gets immediately eaten by the dire wolf, bites off his hand. Amber has to keep running. She finds a car and sort of hides inside that. And of course, she takes a very sensible option of rolling up the windows and locking the door so the dire wolf can't get in. But I mean, I don't think it ever says where this takes place. California, somewhere hot and American.
Actually, it's set in Santa Mira Springs, California, and Santa Mira is it's a fake town that's used in a whole bunch of movies.
Okay.
It's used in Invasion of the Body Snatchers, Halloween 3, most of the Sharknado movies.
Wow.
So it's a standard Hollywood fictional town.
Bad luck place to live if it's got all that going on. All right. So basically, yeah, dire wolf's on the loose. It's killing everyone it meets, apart from that mum and child, and also apart from Tina at the Biogen facility. Somehow she wasn't eaten. Yeah, Jim has sort of got over his disgust at guns and is now carrying a gun with him, and he calls himself a flip flopper. So that's great. We cut to another scene, which is a super sleazy photographer and a bikini model. Your standard Santa Mira inhabitants. It's just an excuse for, I think, some gratuitous butt shots of the very attractive model. And of course, they are flirting, and it's strongly implied that there's a non-professional relationship between the photographer and the model. And the photographer goes to get a blanket and then gets immediately eaten by the dire wolf. And then also the model gets eaten by the dire wolf. So it's really kind of just eating everyone. So the FBI and and Tina from Biogen and Sheriff are out searching for the dire wolf with the classic, let's split up. We'll cover more ground this way. So Agent Reed is with Tina, and the other agent, whose name I can't remember, she is with the sheriff. And they're all looking, and also Jim, the game warden, is looking separately as well. Jim meets the two assholes from the diner who weirdly called him a cream puff, which is it's like a 70s insult, I think. And it's never explained why they call him that. Unless, is it ever explained? But all the way through, everyone calls Jim a cream puff, which is just weird, really.
Yeah, I don't know. By the way, though, these two rednecks, for some reason, everybody feels like they're in the American South, even though it's set in California.
Yeah.
But these two rednecks, I was calling all the way through Roland Shit from Schitt's Creek, if you've seen that.
No, I
and
haven't
Chris
seen Schitt's
Cornell.
Creek.
Oh man, this guy looks exactly like the mayor of Schitt's Creek, Roland Shit.
Okay.
He's it's like he's doing him for Halloween.
All right. So everyone's out searching for the dire wolf. Tina and Agent Reed find the photographer and the model all chewed up. We get a close-up for the first time, I think, of the dire wolf costume and realize it's horseshit. It's a guy in a gorilla costume with like a Halloween wolf mask. It's really not very good. So Sheriff and Agent Reed find the dire wolf and they fire a tranquilizer gun at it. And the agent stops the sheriff from actually shooting it with a real gun. But the tranquilizer has no effect, because obviously that would stop the plot at that point, so they can't have that. Amber is still hiding in the car with the windows rolled up. We get some very kind of Shatner-esque acting of her being very hot and boiling in this car and wondering whether it's safe to roll down the windows and kind of going back and forth over whether it's safe or not, and then finally doing it, and then immediately as it rolls down, she hears she hears noises and rolls it back up. But actually, it's not the dire wolf, it's Jim. He's come to rescue her. So that's great. She's saved. He has to hotwire the car, because obviously he can do that. We meet the rednecks again. They've been kind of told off by Jim, because he's such a good guy that he's told them that it's not safe to be out here while they're drinking beer. He sort of tells them off for drinking beer. I don't know why. They're grown adults. He doesn't tell them off for littering.
Is it illegal to drink beer in this park? Or
yeah,
what?
maybe it's because they're drinking Bud Light. I don't know.
Public drinking?
I don't know. But yeah, so the rednecks are back at the kind of warden station, and it's revealed that these are the guys that did the graffiti that they were talking about at the beginning. So they don't like Jim, which we already knew. But then the direwolf turns up, none the worse for its tranquilizer shot, and immediately eats these two assholes. So that's fine. Jim has hotwired the car, and so him and Amber make it back to the station. He sees the dead rednecks. They both go into the station, and he says, "I'll be right back." And I was sure at this point that he was going to get killed, but no. Tina and her agent get back to the sheriff's station, but immediately the agent gets mauled. But then we sort of Tina reveals, she tells the direwolf, "Stop it," and has some kind of psychic connection, because of course, to the direwolf. And so it listens to what she has to say. But it doesn't matter. The agent that was with Tina is absolutely dead. Agent Reed, who's the senior FBI agent, she is so traumatized by the death of her partner that she now is shoot to kill. And we kind of get more of this psychic link that Tina seems to have with the wolf. And so they're all kind of holding out in the game warden's office. But the sheriff sees that the sign outside the game warden's office is slightly askew. And because it's been established early on that he's, in Hollywood terms, OCD, he has to fix it. So he pretends to go to the bathroom, climbs out the window, and fixes the sign. But of course, he immediately gets mauled by the dire wolf. What an idiot. And so everyone rushes out. Jim stabs the wolf, but the sheriff is dead. We get some touching kind of foster child relationship moments where the sheriff gives Jim his badge and tells him that he has to polish it for 15 minutes. And this is not a euphemism. He has to polish it for 15 minutes every Tuesday to keep it shiny.
At 2:30.
At 2:30, yeah. Tina, thanks to her psychic connection, knows that the direwolf is going back to the lab because it's going back to its den where it feels safe. Agent Reed, the surviving FBI agent, speaks to the army major, who is her boss somehow. Are the army the boss of the FBI? I mean, I don't think so.
Are they FBI? They never really say what they are. They could be Men in Black.
They do have. You get a view of their ID, and it says FBI. I mean, they could have got them from the Halloween shop, for all we know.
I'm pretty sure that's where they got the park ranger, the game warden uniform. Looks like it's got a cartoon deer on the patch.
Yeah, totally. But Agent Reed speaks to the army major and says that she calls him Dr. Moreau, which is probably the highest literary pretension this film gets. And says that they're going to shoot to kill the direwolf, and so the major's upset because this is their bioweapon that they've been developing. They get back to the lab. Their brilliant plan is to just put down a sheet of metal. It looks like the whole fucking building's made of metal.
Yeah, but they put a sheet of metal on a metal floor.
Yeah, it's crazy. So they put the sheet of metal on the metal floor and attach some jump leads to it, and their plan is to electrocute the direwolf, which is impervious to, it seems, bullets and tranquilizer. Anyway, Dr. Renault appears. He is... What does it say? I don't know. I got so bored by this point that I stopped taking notes, really. Oh, yes. Dr. Renault says that they're making more direwolves, and they have like a tiny little jar of direwolf that they're version 2, but Jim is having none of it. He just knocks it to the floor and kills it. And then we get a really tedious spiel about how the thing that the Aztecs were most worried about were not the conquistadors and the guns, but their hunting dogs. And so that's why a direwolf human hybrid is the ultimate weapon. And then we get a bit more exposition, which says that the human donor for the direwolf human hybrid is actually Tina. Her embryos were the source of the stem cells for the direwolf, and that's why they have such a strong psychic bond. Because everyone knows that mothers can always tell their kids to do everything and have a psychic connection to them. But the direwolf turns up. Dr. Renault gets totally mauled. The direwolf gets shot. Tina uses her psychic link to tell the direwolf to stop it, lures it to the plate, and it gets totally fried because Jim and the agent switch the breaker, and that fries it. And Tina's okay, Amber and Jim are okay, and they all say it's time we all went home, and then that's the end of the film. And I didn't watch this at 1.5 times speed because that wasn't an option.
Yeah.
Because otherwise I totally would have.
Well, do we have any inaccuracies we can talk about in this movie?
Any? Well, a big one. So, first of all, when Tina's doing her massive exposition dump, she calls it Canis Dirus, which is not the correct binomial for the direwolf anymore.
And like, you couldn't look that up. You could not Google that. Like, there's no excuse for that now. And what was it? 2009 it was made?
Yeah.
Was it formerly called Canis Dirus?
Yeah, at times it's been called Canis Dirus. Probably in 2009 it was called Canis Dirus. But now it's got its own genus.
Then that's not their fault.
Yeah, but stop making excuses for them, Kim. They're grown adults. They should be able to take the criticism. Yeah, so now it's got its own genus, Anocyan dirus. So, I mean, the paleontologists, the people that first described it, put it into its own genus, which is Anocyan, because they realized from the bones that it was pretty different from the other canids, particularly the Canis genus, which has coyote, dog, wolf, and the sort of wolf-like dogs. But then it kind of got shifted back to Canis for, I don't know, particular reasons. And now with DNA evidence, it's been split off from the Canis genus again into its own genus. So it's like many kind of fossils, particularly ones which we have a lot of bones for. And what am I thinking of here? Well, a good example is the cave lion. So it gets moved around all the time. Some people call it Panthera leo, which is the same genus and species as the modern lion. Some people have given it its own species name, Panthera spelaea. Some people have put it into its own subspecies, Panthera leospelaea, etc., etc. So taxonomy is not an exact science, and it changes with the breeze, and so it is with the direwolf. So yeah, that's one. Other than that, her kind of exposition dump's pretty good. It's the most common fossil at Rancho La Brea. There are more than 3,000 individuals. There has been a lot of discussion about whether it was a hunter or a scavenger. It has really big jaws. It has it's essentially almost like a hyena analog. So there's no hyenas in the late Pleistocene America, whereas the Old World has the cave hyena, the spotted hyena, brown hyena, lots of other kind of bone-crushing scavengers. There's not that in North American Pleistocene. So the dire wolf has sort of moved to sort of slightly fill that niche and has bone-crushing jaws, really strong teeth, really strong muscled jaw, and it's pretty big. It's about a fifth bigger than a big grey wolf. It's its own thing, though. It's outside of the diversity of wolves, foxes, and jackals. It's on its own kind of unique branch, so it's not particularly similar to anything that's around today. But it was a very common species in Pleistocene North America and South America. So, like the North American saber-toothed Smilodon fatalis, it has a similar range. It's not found north of Canada, so it's not found in Beringia, and it's found all over North America and down into the northwest part of South America. So I think the furthest south you get direwolf fossils is Peru, which is about as far south as you get Smilodon fatalis fossils as well. So a similar kind of overlap. And we know they overlap because direwolf are the commonest fossils at Rancho La Brea, and probably the second most common is the saber-tooth Smilodon fatalis as well. So we have huge numbers of fossils from this guy, just from that one site. Yeah.
So Ross, at the beginning, you talked about this folder that she's reading in like the introductory scene that says marrow density on it. And it actually is relevant to the plot because later when we get the exposition dump, we find out that they found this direwolf fossil that was so fresh, the bone marrow was still viable, is what they say. So I think the implication is that there is like literally living marrow cells inside the bone marrow of this fossil somehow. And then they introduced human stem cells to that viable bone marrow in order to create a hybrid.
Yeah.
Do we want to talk about whether or not this process would work?
Well, I mean, so direwolf only recently had its DNA sequenced. So the problem with direwolf DNA is that it's not very common. So Rancho La Brea, despite having thousands of direwolf remains, is a no-go area for DNA survival. So because it's covered in tar, it that just absolutely stops any chance of getting DNA out of it. There might be DNA there, but the kind of experimental techniques that we use to amplify DNA is absolutely shut down by all the weird chemicals in the tar that we just can't get rid of. So I think it was a few years back, the first DNA from a direwolf was actually successfully worked on. I have that paper to hand, which is Perry et al. From Nature in 2020, which has got a lot of people that I've worked with and know on it. It's a good paper. And the direwolf they actually got to work was from a cave site. So there are other sites where you have direwolf fossils, not in such concentration as at Rancho La Brea, but it was these other places that they were able to get viable DNA from. I'm just trying to quickly scroll through and find out where the actual cave was that they got it from. So samples from Idaho and Wyoming, Ohio and Tennessee.
But that is DNA extracted from bones, not from still viable bone marrow.
No, I mean, I've seen bones that have, like from the Pleistocene, that do have marrow in them, but that's from permafrost regions. So
yeah,
when I've done field work in the Yukon and sort of northern Canada, working at the gold fields there, occasionally you'd open up a bone for sampling and find, still kind of red, funky, gross marrow jelly in your bison or horse bone. But it's very dead. It's freezer burned to fuck. It's been there for 20,000 years. There are no living cells in there, but there is, the breakdown products of those cells are still there, and they're kind of held together in a roughly similar way, just by the cold and the breakdown. So yeah, that you're not going to find a living cell from a Pleistocene mammal. Certainly not outside of the permafrost, but even not in the permafrost. That's just not going to happen. When people are preserving kind of living cells in biobanks and cryobanks, you're talking about stuff that's kept at a much lower temperature and in a kind of preservative solution that's going to stop it from forming ice crystals and kind of tearing the cells apart. So, stuff in liquid nitrogen at minus 170, and you just don't find those temperatures in the natural world. So there's no chance of living mammoth cells or living saber-tooth cells being recovered, as they discuss in this not very scientifically accurate film.
If you did want to clone a direwolf or to make a hybrid, you would do that by taking human stem cells and then just introducing them, is what she says. They were introduced to the bone marrow. I don't know exactly what that means. I'm imagining
take them out for dinner.
Yeah, open a bottle of wine.
I'm imagining the scene from Futurama where they get their $300 stimulus checks, and the professor wants to get a de-aging treatment, so he goes to buy some stem cells. He's like, "Is it true that stem cells can combat the aging process?" And she's like, "Well, yes, in the way an infant could fight Muhammad Ali."
Yeah. No, if you wanted to clone a dire wolf topically, which people claim that they have actually done, it's been big in the news this week. Colossal have put out their 3 dire wolves, and this is how they wouldn't do it this way. They wouldn't create a direwolf human hybrid. As far as we know, maybe they're not telling us. Maybe they are just an army front to build bioweapons. But what they've done is they've sequenced the genome of the direwolf. They've identified genes that have been under selection, that influence the phenotype, so influence the living appearance of the animal. And they have taken some grey wolf cells, cultured those, and then used molecular biology techniques to mutate, like selectively mutate the grey wolf genes to make them more like the dire wolf genes. And they've done this in, I think the research says, like 14 different places. Not very many, but still hugely technically impressive. And then they've taken this cell line, which now has 14 dire wolf introduced mutations, and used that to clone an embryo and brought it to term. So it has been kind of hugely controversial. And full disclosure, Professor Beth Shapiro, who is the chief scientist for Colossal, it's a friend of mine. She was my PhD supervisor. I've been a co-author on lots of papers with her. If anyone is going to do this kind of work and do it well, it's going to be Beth. So it feels like a lot of the criticism that this work has received has been not on the technical merits, which are hugely impressive, but on the sort of hype around it. So
it's the media coverage.
Colossal are a business, and however interesting the science that they're producing is, it feels like the business side of it has just steamrollered through everything and hyped it up beyond kind of academic respectability. So my sort of take on it is that it's got slightly out of hand in terms of the hype. I mean, these aren't dire wolves; they're not de-extinct, but they are more de-extinct than anything we've seen so far. So they've done something that's hugely technically impressive, going from living wolf cells, introducing directed mutations that are found in the dire wolf genome, using that cell line to create a viable embryo, bringing that embryo to term and producing, so far so good, healthy pups is incredible. That's just really massively impressive. But 14 mutations, that's not a lot.
No,
and certainly these animals, I think, it is hyperbole to call them dire wolf. They are genetically modified wolves with potentially, I mean, we don't even know for definite that the mutations they've introduced are having any phenotypic effects. Yeah. And certainly there's going to be way, way more differences between the grey wolf genome and the dire wolf genome than 14. I mean, we're talking about a divergent date of about 4.5 million years. So, hundreds or thousands of differences are going to be found between those two genomes, and that's a big difference. But it's definitely got people talking. It's definitely got people thinking. I don't think the kind of negative attention is really based on the science. And negative attention is based on the kind of publicity machine that Colossal has kind of put into motion. And the unsubstantiated claims that they're making without scientific backing, and also kind of pushing ahead with the press release before any paper has been published. So the current work is now on bioRxiv, which is peer review. So no other scientists have had a chance to check the research, but yet for the past week and a half, the news has been nothing but direwolf resurrected kind of stuff.
Mm-hmm.
To me, it seems like I haven't looked at the actual academic work, or even I honestly haven't spent that much time thinking about this, but it reminded me quite a bit of all the hype surrounding like Homo naledi and the Rising Star and like that big publicity stuff. Like, so the stunt of sending up the human fossil into that space shuttle or something. So, it was like I think it spent more time in space than Katy Perry, maybe, but not much longer. And then,
we've all spent more time in space than Katy Perry.
And that same research group, whether you respect their research or not, or whether you respect some of it and not others, they continuously come under scrutiny for making their ideas public and hot and sexy and popular before peer review, right? And even sometimes the Lee Berger kind of pushes back on his Facebook and stuff about peer review not being the be all end all. And then guess what I'm saying is that there seems to be this trend now in science, and especially if there is monetary gain, like commercial gain to it, to skip the peer review process and make the results sound as sexy as possible and then throw them out there. The thing is that like as a minor, non-celebrity, non-doing anything super exciting scientist, I have had my research in media. And when you're talking to someone, like you send out a press release and you have to try and make it sound really interesting, or else it's just never going to, like, what's the point of making a press release? So you'll already feel like you're not skirting the line, because it's all you want to make sure that it's accurate, everything you're saying, but you're selling it essentially, right? Mm-hmm. And then when you talk to the reporters, they, at least some of them, not all of them, but some of them will push and push and push for you to say the soundbite they want you to hear, or for you to confirm the story that they want to tell. And they kind of almost, some of them even get kind of angry and disappointed when you refuse to feed into that, or you refuse to say it or confirm it. And then even sometimes, even after you say, you tell them that that's not the right interpretation and you try and do everything, they still run with that story.
Yeah.
So it's really hard. Sometimes it's the scientist's fault. Sometimes it's not. There's obviously some kind of malignant force pushing this type of stuff, right? Like, so you get the fame or the money or something from it, but maybe we need to, as the scientific community, increase value so that this stops becoming a motivator.
Totally. I mean, and this is the kind of the problem, I think, or one of the problems with science is that it's expensive to do it and to do it well. And where do you get your funding from? So most science is funded through government agencies, through a kind of open tender process where you have to apply for grants. It's very competitive. You have experts that review those grants and only give the ones that sound like they give the best return for the tax dollars invested, which has its own problems, because a lot of really fantastic research is blue sky. It has no apparent benefits at the moment. It's just interesting to find out answers, and then perhaps down the line there will be huge advances made from it, and we just can't tell. I mean, that's the beauty of science. You just never know where it's going to end up. The problem, I guess, with private finance backing it is that you will always get some bias coming into that, whether that's conscious or unconscious, and that's why all papers have to have a conflict of interest statement appended to them. You have to say, when you submit work for publication, you have to state what your conflicts are. And certainly with like Colossal, which is I think like an investment company, like they are looking for investors to put money in with the idea that the advances that they find will have monetary benefit for the investors down the line in terms of them making their money back, plus interest. And the problem is that conservation is not a big moneymaker. Conservation, takes a lot of resources for a lot of intangible benefits. So economists have tried to put a money value on the biosphere or on ecosystems, which to me, as someone sort of almost a conservationist in sort of professional terms, just makes no sense. Because if you don't have an ecosystem, it doesn't matter how much you think it would cost in dollar value. You can't have a dollar value on a dead planet. It just doesn't make any sense to me. So the benefits of investing in conservation are huge, but intangible, essentially. You can't put a price on having breathable air and, a functioning habitats. I just don't think you can. And so how are Colossal going to make money from what is essentially, it feels like, conservation investment?
They're secretly breeding soldier wolf human hybrids, right?
Okay, yes, that would work. Yeah. Hybrid direwolf humans for battlefield use with psychic links to their stem cell donors that can control them.
Yeah, we need to figure out a way of making conservation sexy and exciting, but we've been trying that since the 90s and it hasn't worked, right? So,
yeah. Fundamentally, scientists are deeply unsexy people, and we're suspicious of anything sexy.
Yeah.
And rightly so, because that way kind of leads to bias.
Yep.
And not more than just conscious bias. What's more dangerous, unconscious bias, which you can't control for, and that will affect your results and lead to bad science, which is the last thing we want.
Yeah. It'll make you interpret your results in terms of what you want them to say, not what they say, right?
Yeah, what your funders want you to say.
So to sort of summarize the colossal project here to de-extinct the dire wolf, can we say conclusively they didn't de-extinct a dire wolf? They have just created a transgenic gray wolf. Can we make that statement conclusively?
I think we can, but at the same time, there has been a lot of talk, and Beth talks about this in her book as well, about what is de-extinction. It's something I talk about in my book as well. It's almost a sort of it's a ship of Theseus kind of argument. At what point is something enough like a dire wolf that we would accept it as? I mean, it's gone. It's gone forever. It's extinct. If it had 99% of the same genome as a dire wolf, would it be acceptable? Okay. What about 98%? Where is on the sliding scale? What is acceptable, and why? More importantly, why would it be acceptable at that point and no other?
For me, it would be so like if you take Neanderthals and humans, say, like put it in context that I understand better. For us to say it's Neanderthal and not human. So if we share 99.98 or whatever percent, so there's like that 0.02% that is different from us to Neanderthals, that 0.02% would have to be majority Neanderthal. I would think.
Okay.
So is that the same with but what's the difference between wolves and dire wolves? More than
probably be
foxes and
probably be slightly more than that. But I mean, the issue there is, so there you have your human genome and your Neanderthal genome, and you compare them directly, and you see there's this 0.02% difference. So let's say you make all those changes in your human stem cell line, then there's still going to be differences, because your human stem cell line is not going to be the same as the one that you compared your Neanderthal genome to in the first place. So there will be unique mutations in the stem cell line because it's from a different individual, because it's been sitting in a petri dish for the past 50 years, evolving in its own kind of small way to enable it to survive.
That comes down all the way down into the whole idea of the biological definition of species again, right? So because
absolutely.
Where we draw that line is where we have to draw the line for everywhere. So where is the variation? At what point is it? Those differences in mutation from the time in the petri dish from different individuals? How much is that within what would be expected of mutational variation within a single population or species, and how much, or is it beyond that? Right.
Totally.
And we don't have an answer for that even within living species that we know are different. So I don't know.
It's a really important point as well. One that's not really brought up by the media as well, is that we just don't really have a good definition for what a species is either. It's just like,
no, we don't.
We kind of know it when we see it. There are multiple conflicting definitions of what a species is, and at the end of the day, it's a human value on what is a spectrum. Like, there are no clear boundaries. Everything is a bit fuzzy. Let's say with the kind of group that I'm most familiar with the cats, you can say, okay, lions and tigers, clearly separate species according to how they look. We know that they're different. They live in different places. One's got a mane, one's got stripes. Okay. Let's look at some of the species concepts. Let's look at the phylogenetic species concept. Yes, we have a tick there. So they form their own little branches on the cat family tree when you look at their DNA. So that's great. We can say lions and tigers, phylogenetic species concept, great. But then we look at biological species concept, which says that species are good if they form reproductively isolated populations that can't interbreed. You get a lion and tiger together, they'll breed quite happily. You'll get yourself a liger.
Is it as a liger viable though?
Yeah. So unlike with horses and donkeys where you get a sterile mule, ligers are fertile. You can breed. So ligers are male lions and female tigers. You also get tigons, which are male tigers and female lions. And I'm not sure if tigons are fertile, but ligers definitely are. Then you can also breed them back to lions or tigers again. You get yourself a Thai liger or a Lai liger, or you can even breed them with leopards. You get yourself a lay liger. So, biological species concept falls down on what are clearly good species, because everything is kind of so individual and so unique. Until, I guess, 100 years ago, everyone would have said Neanderthals and humans were totally separate species, but then thanks to genetics, we've seen that actually humans and Neanderthals could interbreed, and we're the end result.
I think just the way we conceptualize species today, phylogeny plays a major role kind of regardless of the species concept. And when we're looking at transgenic species like this, there's nothing built into the concept of species to account for it, right? So, whatever species concept we use, the understanding is that everything shares a common ancestor and they've been diverging for a certain amount of time. And the more they diverge genetically, the more phenotypically different they become usually. But with a transgenic species where you're editing the genome, none of our systems, we don't have a species concept to account for that yet. So that's something we're going to have to come up with in the future to determine if you're modifying a species genetically, how much do you have to modify, or what criteria do you have to determine whether you've created a new species or whether you've just modified the genome of an existing species?
Yeah, that's a good point. We're kind of in uncharted territory. Although, I guess maybe we should be looking to the botanists for this, because obviously transgenic vegetables, plants, cereals have been around for a while. Maybe they can inform us on, or maybe not. I mean, who talks to plant guys?
Well, I was watching a video from Hank Green about this direwolf de-extinction, and one of his points was, he was talking about like transgenic vegetables. He said, we've put fish genes into tomatoes, but that doesn't make it fish. It's still a tomato, right? And so he's saying we've put direwolf genes in a gray wolf, but that doesn't make it a direwolf. That just makes it a gray wolf with direwolf genes in it, right?
But I mean, you could even just go and be kind of annoying and says that this entire argument is a human construct, and so we can define it any way that we agree upon, right?
Yeah.
So,
but that's getting humans, particularly scientists, to agree is not an easy thing.
If they all gave us money, though, to say that was a direwolf. Maybe.
Yeah, just putting it out there. If Colossal wants to throw money at us, we'll say whatever they want.
Whatever. The next episode of Screens of the Stone Age, sponsored by Colossal Biosciences.
Yeah, if there's a steady paycheck in it, I'll believe anything you want.
That is not true to our listeners. If that were true, we would be making more money than we do.
One thing I wanted to ask you about here, Ross, we can agree there's definitely good, real, important science going on, and it's the hype that we are mainly criticizing here. But that hype sometimes is coming directly from Colossal themselves, because I'm looking at Colossal's page on the direwolf here, and it's super flashy. It's a really well done, graphically interesting page. But it's talking about why it's been so hard to get DNA from the direwolves. And it's talking about them being mostly the ones from La Brea aren't viable because they've been in tar. And it says here, this high heat environment allowed the organic molecules of La Brea's tar to penetrate bones, dissolving and destroying usable DNA. But hot tar isn't entirely to blame, blah, 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 blah. We've talked about the tar pits before. They're not hot. It's not the heat that's the problem in the tar pits, is it?
No. Though, California is hot, and it's not good to have 40 plus temperatures for DNA preservation. The problem with the La Brea, the preservation is good, the biological preservation. So the collagen is great, and that's why there's lots of radiocarbon dates from La Brea, because the collagen is well preserved. The problem with the La Brea bones for DNA work is that the tar itself is such a kind of heterogeneous mixture of really odd organic molecules. You have all sorts of stuff in there. And that really interferes with the quite delicate enzymes that we need to use to amplify DNA. So the DNA polymerase is inhibited by lots of things, including all the unknown stuff that you just can't get out of the bone because it's all the way through there from the tar pit. So certainly the heat of California doesn't help. It's not the heat of the tar; it's the heat of California. But more than that, it's all the unknown chemicals that have sort of seeped into the bone from the tar pit that we can't get rid of without further breaking down the DNA. That's what makes it so impossible to work with. And this has had direct relevance on my kind of academic work. So back in 1996, there was a big paper from a lab looking at DNA from Smilodon, the saber-toothed cat from La Brea. came out in PNAS, so a big journal, Jansuski et al. And their claim in this paper was that the saber-tooth was part of the modern cat radiation, so part of the Panthera genus, the same genus that has lion, tiger, leopard, jaguar in it, which was a huge claim because up until that point, all paleontologists had put the saber-tooths, and Smilodon in particular, in their own subfamily, the Machairodontidae, which is totally separate from any of the modern cats. So it was a big claim. And it was high profile and published in this journal using DNA from a bone from La Brea. And one of the first things I did when I was working on my PhD was to look at this gene sequence that they'd produced for this paper and see what the hell was going on with it. And it was a very small fragment. But what was very obvious, even before I had any DNA, real DNA from a saber-tooth, was that this was from a domestic cat. What they'd amplified was DNA from a domestic cat. And because of the way that phylogenetics works, where you have to align your sequences to sort of maximize their similarity, what they'd done in this paper was aligned it badly and not in a parsimonious fashion. And so that allowed this domestic cat sequence to be read as part of the Panthera branch. And that paper's never been retracted. I mean, everyone knows it's wrong. And there's been so many papers that have come out saying why you're not going to get DNA from La Brea specimens, but it still gets cited occasionally.
Ross, maybe I can ask you a little bit about what is the value of de-extinction for conservation? Because I'm sure that there is, but in this particular case, is there, I mean, this is experimental research where they're doing like proof of concept stuff, which is what's really valuable. But is there any good reason to reintroduce dire wolves to North America, considering that the ecosystem that they evolved in doesn't exist, and that's why they went extinct, right?
Yeah. I mean, this is something that I talked about in my book, Beth talks about in her book. The sort of short answer is no, not really. Longer answer is slightly more complicated. Certainly, if we're talking about sort of de-extinction of species, then you wouldn't really want to start with the predators. There is, I think, value, or there could be value in bringing back some of the herbivores because of their huge influence on shaping the habitat they live in. So when we talk about, parts of Siberia, for instance, which are now sort of tundra taiga, very unproductive ecosystems, and very different from what they were like during the last ice age when they were kind of productive mammoth steppe, a bit like the Serengeti or the Masai Mara, places like that, really kind of packed full of biodiversity, biomass. And a lot of that is due to kind of the interaction, the sort of symbiosis between the environment and the animals that live in it. So when you think about places like the Serengeti or the Masai Mara, all these other kind of intact ecosystems in Africa, they have a functioning megafauna. They have elephants, rhinos, hippos, zebra, giraffes, all these other huge mammals, and they are modifying the habitat to keep it the way that it is. It's a delicate balance, and so you have your elephants pushing down trees and creating open areas in which grasslands and herbs can flourish. You have hippos wallowing in the mud and kind of moving. So hippos feed on water plants, but then they come on land at night and eat grass as well and defecate. So they're moving kind of nutrients around between different ecosystems, from kind of riverine to land ecosystems. So you have this incredibly complex interactions, which are now totally lost. And so, whereas in the Pleistocene, the kind of region of Siberia to Alaska, the kind of Beringian ecosystem was, a flourishing place where you'd find lots of megafauna, lots of diversity. Now it's a lot less diverse and a lot less resilient as well, because there aren't these kind of checks and balances in place that would help to kind of keep things moving along. So I would say that there can be a real net conservation benefit if, and there's lots of ifs involved with this, if you're able to bring back some of the herbivores to such an extent where they were able to reintroduce their original natural behaviors in modifying the environment as they used to. There's not a lot of relevance in bringing them back just to keep them in a zoo for people to kind of gawp at. I would be very much against that. But it's tricky. And yeah, certainly direwolves is not where I'd start. And even if we were to try to bring back more of the kind of ecosystem engineers, there would be lots of questions about whether it would be a good idea to put them back into habitat, which is different from what they had, to see if they could bring back the interactions that used to be there. Yeah, it's tricky. And I don't really know the answer. I mean, the cautious part of me wants to say that it's just vanity. Certainly with I mean, it depends on the de-extinction project as well. So, like, Colossal are also working on the dodo and the thylacine, and I would say for them, these are 100% human-caused extinctions within the past few centuries, within the last century, in the case of the thylacine. And there's a much stronger argument for bringing them back and just saying, right, we've got them back, put them in the landscape, which hasn't changed much since they were there. Put the thylacine back in Tasmania, put the dodo back in Mauritius, and then just sort of let them be, see what happens, and that would be great. But because we're still not, there's still not a consensus on the extinction of the Pleistocene megafauna. I think it's a much more difficult project to sell.
One of the other applications of this technology, I believe, is that they can create more genetic diversity within living taxa, which would be very good for conservation, because that's going to help species that are close to extinction not have genetic problems, right?
Yeah, and I think they're actually doing this at the moment. Like, I think there was a press release within the past few days where they talked about reintroducing diversity into, was it red wolf? I don't know if you've read this, so. Josh. Yeah. So yeah, absolutely. That is a positive aspect of this sort of science that could help conservation. And I think they're also doing that with black-footed ferret as well. Again, another close to extinction American species that is now not very diverse. I think the problem there where you'd have to be careful is that if you're doing the same kind of route to introduce diversity into things, you'd have to. It's almost in the same sort of category as when you take the last members of a population into a zoo to do kind of ex situ breeding to increase the numbers and then re-releasing them back. The time in the zoo is going to affect the individuals because they're not in their kind of wild natural environment. In the same way that if you're taking sort of cells out of your red wolves or your black-footed ferrets and putting them in the petri dish in the lab and letting them reproduce and divide, and then kind of modifying the genes to introduce the diversity, that is also going to have an effect that may be unquantifiable. They may pick up mutations that you wouldn't want them to have down the line, just because we can't control stochastic random mutation that happens just to everything all the time.
Yep.
Cool.
So yeah, it's much easier just to not let things go extinct. I think that's probably the take-home message.
You should do a plug for your book, Ross, for our new listeners.
Yeah, read my book, buy it.
The Missing Links, LY
know it.
and X.
Yeah, and I do talk a bit about de-extinction and the ethics and quandaries in there as well. And
Beth Shapiro's book, How to Clone a Mammoth, is good too.
Yeah.
All right, well, we've been talking for over an hour.
Ross has been talking for over an hour.
Yeah, true.
All right, Ross.
I need a drink now.
Yeah.
What are our final thoughts about the movie Direwolf from 2009?
I mean, it sounds the same as the saber-tooth ones and the Neanderthal ones. Like, it's all the same horror movie thing. This thing from the past gets brought to life. It goes on this monstrous rampage until it's taken down by some crazy whatever.
Yeah, by the power of love.
Yeah, man and woman fall in love.
Stem cells.
The guy gets a date. It's all,
It's funny that in this one, Tina thought that their research was supposed to be curing cancer, and I'm pretty sure that's what the goal was in Sabretooth as well, to resurrect the Sabretooth for some way they were going to cure cancer by resurrecting a Sabretooth, and I don't know how that worked.
It's very much underpant gnomes, isn't it? It's step 1, clone your dire wolf. Step 2, question mark. Step 3, cure cancer.
One thing this movie has going for it is a lot of blood. If you like Lots of blood and gore. They put probably 50% of their budget for this movie into fake blood.
Yeah. More than they put into the costume, I would say.
Or the plot.
Yeah. Or the acting.
Do you have questions, comments, or movie requests? We want to hear from you. Find us on Blue Sky and Facebook at SOTSA Podcast. Send us an email to screensofthestoneage@gmail.com or visit our website at screensofthestoneage.com.